Okay, yeah, so I would like to thank the organizers for making such a great meeting, and thanks to assessors for looking into this new category. And I can imagine it's very challenging. It was very challenging for us too, because originally, like with all these formats and everything, was pretty tricky, kind of like to get in the mode. But yeah, it was pretty interesting. So our interest in ligand modeling actually started also around the time when D3R meeting started. So we decided to participate. And at the time we felt that we didn't want it just to use kind of like what was there. I think one round we did use it and we were not super happy. Like even for template-based modeling, like the tools were not doing exactly what we want. So we decided actually to do some kind of like something of our own. And kind of that's the protocol kind of which we mostly used in this was ClassPro LigTBM, which by the way is basically is available and you can use it online. I mean, we actually now deploying a new version and adding new things. So for for one target, like which basically was like there was no good template, like we're in process developing another automated docking protocol, which basically I can will also describe. So, but most of the results coming apart from this one target are coming from the LigTBM protocol. So basically, so what does it do? Because in principle, template based, you can think, I mean, okay, I mean it's simple. I mean, you basically like take a template, copy it, and do the ligand and do the protocol. But the issue is that with protein ligand interactions, as was already mentioned with assessment, there are all these issues with basically with symmetries, kind of like with atom types and everything, that even making like good template based protocol, right, is also takes some challenge. But I think like there is some consensus in community, like what we saw in D3R, and looks like here that like best methods are using like more or less kind of ideologically same ideas, right? I mean, like so first like key is basically what you're trying to look at, like is a keyword here is a maximal common substructure. So basically what you're trying to find like something what exists in your ligand and also in the template. But then turns out to be like there are many ways kind of how you can do it. I mean, like in terms of like alignment to the target protein structure, also how you look at the multiple common substructure, whether it's like global, whether it is strict or kind of like, or more relaxed, right? Because strict allows basically like gets you more reliable models, but relaxed like VCMCS give you actually like a better, I mean, like can pull things from very remote template. So basically kind of the idea, like what we do here is like you take a ligand, identify if there is a maximal common substructure to a target, take our original ligand, very exhaustively sample it. It's around like 10,000 conformations, sometimes even more, and then try to fit all possible ways to the target protein. Then the target protein could be either derived like from your provided structure. So in this round, we mostly used AlphaFold based structure. So if it's a monomer, we prefer to use them from the AlphaFold database. If it's a multimer, we basically use the multimer, because that also one of the things which we wanted to see how AlphaFold models perform for docking. So another kind of interesting things in terms of sampling, what we do, like for linear molecules, we use like RDKit with ETKDG, but for macrocycles, actually, we use like more specialized macrocycle sampling algorithm called Bricard, which is developed by my colleague at Stony Brook University, Evangelos Koutsios. And this algorithm is actually like, I think by now, like almost everywhere in Rosetta, everything. So this is kind of a pretty good thing if you need to sample macrocycles. Okay, so yeah, and then I guess also we have like some special local optimization algorithms. for the thing. And then finally the scoring, where again you basically most of the scoring like tries to choose the models which are staying as close as possible to original template in the region where the template exists, and basically in other places it exhaustively samples because of full conformational sampling, so you can actually choose like the best possible conformation. And so just to see an example how kind of how does the steps of the protocol help, right? So the initial sampling gives you actually pretty decent this is example from some previous D3R experiment. It gives you pretty decent kind of like RMSDs, kind of like 2, 1.5, but like local optimization actually can push it even lower and basically get you like pretty good structures. So regarding the free docking, like what basically our twist on it, right? I mean, like we didn't, I mean, yeah, we're developing some of the docking programs, right? But it's not ready for the prime time for ligand docking, but actually for the direct ligand docking. But one thing what we use is actually, because there are two questions with using direct docking. One is a box definition of the box, and another is basically how you score the compounds, because even the best like energy functions like from Glide like and Vina are not always perfect. I mean, especially if you focused on one particular pose. So as additional filter, what we're using is actually is something called hotspots. That for that one we actually do develop an approach. So basically what this method does, it takes a protein and like models interaction. It sort of docks with a small fragment size molecules and tries to see the patches on the surface which binds all the different fragments at the same time. So the idea what you would get is on the protein structure, it gives you kind of some locations on the surface where like several probe binds, and from one side it gives you location of the potential boxes for docking, and another thing, like once you have your poses, like what we have the observations that actually good ligands typically want to agree with with this hotspot, so that can use especially like as an additional filter for placement. Yeah, and since there is actually a server for FTMap, yeah, we didn't do a server yet actually, which does which actually integrates it, but it's something which is in process. So again, for CASP we used we docked mostly to AlphaFold models. So yeah, I mean, like in template-based protocol, protein structure plays role, but not, I mean, like not as big, right? I mean, like as long as there is like rough similarity of template and structure. Actually, before AlphaFold, we actually used building models based on particular templates. I mean, like, and I think for one case we actually like did that, like, but we didn't do it systematically. So we just took like a raw AlphaFold models. For docking, like to seek some consistency in docking, we use actually docking to several structures which are most diverse, as that was on this one example. So just to go through briefly, like I guess, yeah, like there will be many iterations or versions of this, right? I mean, I think that what kind of got interesting to us, like there was one case which was declared as kind of monomer, but it was clearly like that it has to have 2 units basically for stabilization of the ligand. And if with absence of that second unit, which basically wasn't asked to be modeled, the ligand like would be badly placed. So we did report it only 1 unit as suggested, but basically modeled it with 2. So yeah, like with this bigger with the bigger assemblies, like I mean, like it seems like again there were good templates. I guess the challenge was only to figure out what is ATP or like AGS there, and basically since without better knowledge, I mean, since there was several models, we just had the one model and we're doing the possible permutations. So yes, this case was interesting. Like the templates was pretty limited. It was antibiotic to beta lactamase, because actually like there was like I mean there was basically like quite a few templates. By the way, I'm not sure, is this compound covalent? I think at least like all the templates were covalent, so it might be like, I mean, not covalent through cysteine, but processing. But yeah, what was interesting, there was a pretty robust template of part of the ligand, but the rest was hanging, and there was some very suspicious and deposited structure, basically deposited structure, but without no publication of this compound, which was binding totally incorrectly. So we decided to trust basically that and it was a homologous protein. So basically, if you would follow the template, things would get really kind of messed up with the protein structure. So we decided to trust more or less the structure, and actually like it resulted in a good high quality model. So I guess this was interesting in terms of question of conformational sampling, like because there is this one case with ATP, like, and as Pat mentioned, like most of the kind of docking of the original ligands, which basically required free docking, were done into the open structure, and that structure was closed. So in our experience, so when we try to take like all the AlphaFold models from PDB were in the open state. So basically, like what we did, even when we provided a template of the closed channel, like actually like MSA was still basically overriding it and basically forcing it to be open. So basically, what we did, we reduced actually like to a smaller MSA, and then it basically generated a reasonable structure, but with closed. I guess it's to the discussion of multiple conformation sampling. So yeah, like so things like not always went well, right? I mean, like for example, some of these longer cofactors, like the tail basically like went the wrong way in the template. And interestingly, I didn't realize, because in the ligands, in the submissions, there was something which is called relevant yes and non-relevant no. So we assumed we don't need to model those. But then I saw actually some people did model. So what was the intended thing? Was it kind of, should we model everything or not? I guess it wasn't very clear in the description. So, because there are actually several other ligands which could be affecting the results, right? So another kind of case is, again, there is some protein conformation where we get like roughly like the right places, but ligands are a little bit off. So regarding the docking, so docking with ion channels, we did have already some experience, because we participated in a GPCR doc, which was organized by Ray Stevens in actually like it was last fall, and this assessment was done in April. And we did actually, like it was also same problem, docking like a ligand to transmembrane protein, obviously to GPCR. And what we found, basically like the conclusion was that we were able to dock with our protocol like the structure, and the resulting RMSD was basically like, usually because in the ligand field people consider if it's under 2, it's considered reasonably good. Like, I mean, like what we got there was like 2 point something. It was like one of the top predictions in GPCR doc. But yeah, it wasn't kind of crystal quality. So basically we still didn't have anything better. So we followed this protocol, and results were kind of similar. So I guess that's basically the accuracy you can expect from docking to AlphaFold models. So it was interesting that docking to multiple conformations kind of like supported like the primary pose, in addition to agreement with with other things, right? So another compound didn't work, right? I mean, like, although it did work for others, I guess they used something differently. So I guess in summary, like the templates kind of like the CASP targets like were dominated by existing templates, which I guess is natural, right? Because basically these targets like are usually kind of some natural cofactors, ions, like things like that, right? And there is a lot of information on this in PDB. And so, but there were some kind of cool and interesting and challenging targets. So like, yeah, so it was interesting, and something was really difficult, right? And then I guess the conclusion, like, yeah, so definitely it can be used, like AlphaFold models can be used for this template-based modeling. I mean, and even for docking, but at this point the accuracy kind of is reduced. So I guess on this, yeah, I would like to thank kind of people from my lab who worked on it. So the main person actually like is Sergey Kotelnikov. So basically like he's a developer and kind of like main kind of like, yeah, like basically he takes the most brunt of the effort working on this. Andrei Alexeyenko, who helped with developing of the methods. Then Mikhail Ignatov and Dmitry Podhorny, also collaborators, Vageli Kutsias for macrocycle collaboration. I mean, yeah, here it had less important role, but I think like for some challenging targets, it's really good to have it. So like, and then Alex Tropsha and Konstantin Popov kind of, so like on help with basically like with doing some of the docking for us. And so I guess that's kind of like it. So I would like to thank the funding and again the organizers. So thank you very much.
Models, did you consider the reliability of the models, the PLDT scores from the AlphaFold, or was that not really an issue relative to these active sites?
I mean, for this kind of like, I mean, targets, like, I mean, like we knew that, I mean, like in the pocket where basically like I guess the most important thing was for direct like physical like based docking. So in the main side, from one side, confidence was pretty high, although we knew that the protein is moving because it's a like transmembrane